Welcome to Stephen classes. Today we have part 7 of human reproduction. We are going to discuss in detail fertilization and implantation. So we are going to begin our class right now. Ready? Oh, coronavirus! Please go from here. We have some serious business. Please don't disturb. Please go. Oh my God. Oh, thank God. It has gone. Now we can continue with our class. Now we have topic, as I told you, fertilization and implantation. So here, going to the first slide, we are going to talk about fertilization. Are you all ready? Okay. So what is fertilization? Fertilization is a process by which sperm cells and egg cells they join. So sperm enters into the ovum and that results in fertilization. For fertilization to occur, there should be insemination. As you know, in the reproductive events we discussed earlier, I have given you an idea about all the reproductive events. So here, first of all, there should be insemination. And how insemination occurs? Insemination will happen only by copulation or coitus. We can call it as a sexual intercourse. So by that process, the semen will be released by the penis into the vaginal region. So that process is called insemination. So insemination will introduce the sperm cells into the vaginal region, right? Now, from the vaginal region, sperm cells they are going to travel. They are motile. They can swim. So from the vaginal region, they will go to the cervical canal or to the cervix, and from the cervix, sperm cells will travel to the uterus, and from the uterus through the isthmus region they will reach the ambulary isthmic junction. So this is the way in which sperm cells are moving from vagina to cervix, from cervix to uterus, from uterus to isthmus, and from isthmus to the ambulary isthmic junction. The junction between the ampulla and the isthmus is called ambulary isthmic junction. And what about the ovum that is released from ovary? Ovum which is released from ovary, it will be here. So these ova or secondary oocytes, they will be received by the fimbriae, and from there ova will reach the infundibulum. So there will be only one ovum released during a menstrual cycle or during ovulation. So that ovum, it will be reaching the infundibulum, from infundibulum to ampulla, and from ampulla it also has to reach this junction called ambulary isthmic junction. And only when both these sperm cells and ova they reach simultaneously at the junction of ampulla and isthmus, there will be fertilization. For fertilization to occur, the ovum and the sperm cells they will be transported simultaneously. They have to be transported simultaneously to the ambulary isthmic region. Then only fertilization will occur. So here in detail, the path of sperm cell is given. The motile sperm cells they swim rapidly and they pass through the cervix, they enter into the uterus, and finally they will reach the ambulary region of the fallopian tube. Then what about the ovum? As I explained, ovum will be released by the ovary. It is also transported to the ambulary region where fertilization takes place. So we have the path in the next slide. So in this next slide, you can see how the sperm cells are moving. They are moving to the vaginal region by insemination. From vagina, they will be transported to the cervical canal. From cervical canal to uterus, and from uterus to the isthmus region. Okay, and what about the ovum? Ovum, which is released from ovary, it will go to the fimbriae. Then there will be infundibulum, from infundibulum to ampulla. And both of them simultaneously, these sperm cells as well as the ovum, they have to reach this junction. Simultaneously, they have to reach the junction called ambulary isthmic junction. Then only fertilization will happen. So the sequence of sperm cells and ova very well represented here. You have to keep in your mind the correct path of sperm cells and ovum. Now, to know more about the fertilization, you should have a clear idea about structure of the ovum. So here you can see in this picture an ovum. Ovum is having a haploid nucleus. It will have a set of chromosomes. 23 chromosomes will be there in that haploid nucleus. And outer to this nucleus, there will be a cytoplasm. You can call it as ooplasm. And you can also see a membrane. That membrane covering the ovum is called ovarian membrane, or it is a plasma membrane. Outer to that, there is a space, there is a narrow space. You call it as the perivitelline space. Perivitelline space. Then you can see a covering, a non-cellular covering. In the beginning, I told you about the secondary oocyte covered by zona pellucida. So here, the secondary oocyte is covered by zona pellucida. So here you can see zona pellucida retained here. So this is zona pellucida. It is a non-cellular layer made of glycoproteins. And outer to that, you can see some radiating cells of, you can call them as the follicular cells, which form corona radiata. The follicular cells, they form a layer of cells called corona radiata. The cells are radiating like a sun. That is why we call it as the corona radiata. So these are the parts of ovum. You will have a haploid nucleus, ooplasm, ovarian membrane, perivitelline space, zona pellucida, and corona radiata. For fertilization to occur, the sperm cell has to penetrate through the different layers like corona radiata, zona pellucida. So only then it can enter into the ovum. So to know more about this, we have the process of fertilization represented here. First of all, for fertilization, the sperm cell has to come in contact with the zona pellucida layer of the ovum. Sperm cell, first of all, it will come in contact with the zona pellucida layer of the ovum. So here, when it comes in contact with the zona pellucida layer, there will be some changes happening to the membrane. So the ovarian membrane, it will undergo some changes, or it will undergo a thickening. There will be some reactions happening inside the ooplasm. There will be cortical granules, and there will be some reactions, and these reactions will result in thickening of the membrane. The membrane is getting thickened, and now additional sperms cannot enter. So after making a contact with the zona pellucida, now additional sperm cells cannot enter into the ovum. So only one sperm can enter into an ovum. So that is called monospermy. So here entry of more than one sperm will be prevented. Okay. The thickened membrane, you can call it as the fertilization membrane. So actually the formation of fertilization membrane, it prevents polyspermy. So when fertilization membrane is formed, it will prevent polyspermy. What is polyspermy? Entry of more than one sperm. So only monospermy will be allowed here. Only one sperm is able to enter because there is thickened membrane or fertilization membrane. So this is the change happening when a sperm cell is coming in contact with the zona pellucida layer of the ovum. Is it clear for you? Okay. So monospermy is allowed. Polyspermy is prevented by formation of fertilization membrane here. Right. Now, in addition to that, secretions of the acrosome, they play a very crucial role in the penetration of the sperm cell into the ovum. The secretion of the acrosome will help the sperm cell to enter into the cytoplasm of the ovum. So, within acrosome, you have a collection of enzymes. You can call it as sperm lysins. So, these sperm lysins will penetrate through the corona radiata, zona pellucida, and finally it will penetrate the plasma membrane to reach the ooplasm. So here sperm glycines or acrosome, it helps in the entry of the sperm into the cytoplasm of the ovum. You got it? So acrosome plays a very crucial role in penetration of the egg membranes, mainly the layers of corona radiata and zona pellucida. Right. So now fertilization occurs. Now in the next slide you can see it over here. Sperm entry induces the completion of the meiotic division. So once sperm enters, the cell cycle will restart. It was in the metaphase stage. So once the sperm enters, it will induce the completion of the meiotic division of the secondary oocyte. So secondary oocyte will complete the second meiotic division. That means there will be liberation of the second polar body. So second polar body will be liberated and the cell transforms into a haploid ovum or oocyte. So here, only after the sperm entry, there will be completion of the second meiotic division and there will be formation of a second polar body and an ovum. You got it? So the haploid nucleus of the sperm, now you can see two haploid nuclei within that cell now. There will be haploid nucleus of the sperm and also there will be haploid nucleus of the ovum. Both of them will fuse. Both the haploid nuclei will fuse and that will result in a diploid zygote. So the cell is now getting transformed into a diploid condition and that diploid cell you can name it as the zygote. So this is now completion of the entire fertilization process. So don't think that once sperm enters, fertilization is over. Fertilization will be over only when the two haploid nuclei fuse to form a diploid nucleus or a diploid zygote. So fertilization process is over by all these events. Now about sex determination in humans. In the case of human beings, we are dioecious, male and female individuals are there, and we have difference in the arrangement of chromosomes. What is the arrangement of chromosomes in male and in female? In the case of male individual, you can see there will be 44 autosomes and X chromosome and Y chromosome as sex chromosomes. Okay, so there will be 44 autosomes out of the 46 chromosomes. 44 autosomes and 2 sex chromosomes will be there. One will be X chromosome and other is Y chromosome. Whereas in female, there will be 44 autosomes and 2 X chromosomes. So that is a condition. Okay. So in male, sex chromosomes are X and Y, whereas in female, the sex chromosomes are X and X, or 2 X chromosomes you can see in female. Now, when there is a formation of the gametes, during the gamete formation, there will be a reduction division or a meiotic division. So in the case of male, when the sperm cells are formed, what is the result? In the case of a male, when sperm cells are formed, the sperm cells will have 22 autosomes in them. Is it right? And 50% of the sperm cells formed in the case of a male will contain X chromosome and another 50% will contain Y chromosome. So half of the sperm cells in male produced will contain X chromosome and the other half will contain Y chromosome. So two types of sperm cells are formed in male individuals in the case of we human beings. Okay, one set or 50% with X chromosome and 50% with a Y chromosome. What about a female? In the case of human females, all the ova produced will contain X chromosome only. Am I right? All the ova or 100% of the ova will contain X chromosome. So here, when the fusion happens or when fertilization happens, sperm enters into an ovum. So here, the type of the sperm cell entering into ovum is critical because when a sperm cell carrying X chromosome is entering into the ovum, what is the result? then the result will be a female because XX condition will be produced. So it will be a female. So when a sperm cell with the X chromosome enters into an ovum, then the result will be a female. When a sperm cell having Y chromosome, suppose this sperm cell is going to enter into this ovum, then what is the result? The result will be a male individual. So here, the type of the sperm cell entering into ovum is important. So here, the male partner is having the important role in sex determination. The male individual or father is responsible for sex determination in the case of human beings. Is it clear? Because the father or the male partner will produce two types of gametes. Okay, but the female or the mother will produce only one type of gamete. So that will make the difference here. Okay. So who is responsible for sex determination in the case of human beings? It is the male partner or father. So scientifically, it is correct to say that the sex determination or the sex of the baby is determined by the father and not by the mother. So this statement is true here. Okay, so that is about sex determination. I think everything is clear. To know more about the embryonic development, this slide is very much helpful. So here you can see a secondary oocyte. To the secondary oocyte, the sperm enters, and that event is called fertilization. And after fertilization, a zygote will be formed. Zygote is converted into a two-cell stage. Now the two-cell stage is transformed into a four-cell stage. From the four-cell stage, it will be divided into an eight-cell stage. From eight-cell stage, it will be converted into a 16-cell stage. Remember, 8 or 16 cell stage can be called morula. So 8 or 16 cell stage, you can name them as morula. And morula, further it undergoes cleavage and it will be converted into a blastula. You know, blastula is a structure which undergoes implantation. So all the cells which are formed as a result of this cleavage process, you can call these cells as blastomeres. These cells which are formed, they are called blastomeres. Okay, so the sequence is very important. Fertilization will result in a zygote. Zygote undergoes cleavage, and by cleavage, there is formation of a morula, and this morula further get transformed into a blastula, and this blastula undergoes implantation. Is it clear for you? Now we have next slide, which shows all these structures again. The zygote, cleavage happens, a 8-cell stage is formed, cleavage again happens, blastula is formed, and blastula, it will have a cavity inside. What is that cavity called? Blastocele. The cavity inside the blastula is called blastocele. So this is a cross section of the blastula. Blastula is a structure which undergoes implantation or attachment of the embryo to the uterine wall. It will happen to this blastula. And this blastula, it further get transformed into a gastrula. Blastula, further it is getting transformed into a gastrula. Okay, gastrula will have the germ layers. It will have ectoderm, endoderm, mesoderm. All these germ layers are formed within the embryonic structure called gastrula. So this is the sequence of events happening during embryonic development. So during embryonic development, I repeat, the zygote undergoes a cleavage, and as a result of the cleavage, a morula will be formed. Morula is getting transformed into a blastula, and blastula further it will transform into a gastrula. So this is a sequence of events happening during embryonic development. And one more thing, during this embryonic development, there will be movement of these embryonic structures within the oviduct. From the ampulla region towards the uterus, these embryonic structures are moving. To know further, we have the picture for you. So in this picture, you can see the ovary over here. So you can see the oviduct. Can you see the oviduct here? Yes, this is ovary. You can see the oviduct portion and you can see over here the uterus. Right. So the embryonic structures, they are arranged here in their correct sequence. So here, when you consider this is a ovum, when it undergoes a fertilization, where these structures you can locate in the genital tract, where you can see them. Ovum, you can locate that ovum over here in the infundibulum. Because after ovulation, fimbriae, they are going to receive this ovum. Fimbriae are the structures going to receive this ovum, and this ovum will be located over here in the infundibulum, the funnel shaped part of the oviduct. And what about fertilization? Fertilization is an event happening in the ambulatory isthmic junction. So fertilization event labeled as B, you can see it over here. Is it right? Is it okay for you? Now, the two cell stage, after fertilization, a zygote will be formed and it is converted into a two cell stage. Where you can see the two cell stage, you can see it here. Okay. So now it has started its journey from the ambulatory region towards the uterus. So from the ambulatory region after fertilization, the embryo is slowly moving towards the uterus. So when the development happens, it will go to the direction of the uterus. Okay. Now here the four cell stage, you can see it over here. The 8 cell stage, this is 8 cell stage labeled as E. You can see it here, right? Can you see all these structures within this fallopian tube? Right. Now, what about these morula? Yes, this structure which is called the 16 cell stage, you can call it as the morula. F is also, E is also morula. 8 or 16, they are called morula. So here, F, you can see it over here. Okay, so here you can see the F or morula structure with 16 cells and the blastula, the structure which undergoes implantation. It is labeled as G here and this blastula you can see it over here. So now when it forms the blastula, 7 days after fertilization, a blastula will be formed. A blastula will be formed 7 days after fertilization. So it will take 7 days for this journey. 7 days for this journey for the embryo to reach the uterus. So within the uterus, now the structure is called blastula. So this blastula, here you can see it undergoes implantation. So which structure undergoes implantation? It is our blastula. Is it clear? So for question or for exam point of view, they will be giving some of these structures and they will ask you to locate them in the correct position or locate them in the fallopian tube. So you will have to identify the location in the fallopian tube correctly according to the pictures given as your question, given in your question paper, right? So now we will have some points regarding this embryonic development. The mitotic division starts as the zygote moves through the isthmus of the oviduct called cleavage and it will proceed towards the uterus. The embryonic structures, 2 cell stage, 4 cell stage or 8 cell stage. These cells are called blastomeres and they are formed as it moves along the oviduct. And the embryo with 8 or 16 blastomeres, we call it as the morula. So I am repeating everything. Okay. Now morula continues to divide and transform into a blastocyst. And blastocyst as it moves further into the uterus. Now we have the structure of blastula here. This is very important. So this picture is very important. Here we have the picture. In this picture you can see the outer layer of cells. The outer layer of cells, you can call it as the trophoblast. What is the term given for the outer layer of cells? Trophoblast. And you can also see an inner cell mass. You can see a cavity for the blastula. Cavity of the blastula is called blastocele. So these are the things labeled here. And you should know the role of inner cell mass and also the trophoblast. Inner cell mass it gets differentiated as the embryo. Inner cell mass will develop into the embryo proper, right? Then what about the trophoblast? Trophoblast layer, it will get attached to the endometrium and it will develop into the fetal membranes. So which part of the blastula will attach onto the uterus or attach onto the endometrium? It will be the trophoblast. Trophoblast attach onto the embryo, onto the uterus. Is it clear? So this diagram is important and also the function of these two parts are very important. So we have the implantation now. What is implantation? Implantation is not merely attachment of the embryo to the uterine wall. Suppose this is blastula. When blastula attaches, you cannot merely call it as an implantation. After attachment, the uterine cells will divide. After attachment, the uterine cells will divide and it will cover the blastocyst. You can see it over here. The uterine cells, they divide and they will cover the blastocyst, and the blastocyst finally it becomes embedded within the uterus. The blastocyst becomes embedded within the uterus. This is what is called implantation. So by implantation, we mean not only a mere attachment of the blastocyst. So after attachment, the blastocyst becomes embedded in the uterine wall. By the growth of the endometrial wall, the blastocyst will become embedded in the uterine wall. Then only we can call it as implantation. Is it clear? So with this topic, we conclude part 7 of human reproduction. So fertilization I have explained in detail, and also the embryonic development and implantation I have explained to you in detail. So it's goodbye from Stephen classes. I will come with another part 8. In part 8 we are going to discuss about placenta, about the fetal development, about parturition, lactation, etc. So see you in part 8. Goodbye.